of this Responsible Conduct of Research seminar series. So I have a couple of announcements, and then we'll get into it. So our classes are from 3 to 4 on Thursdays through October the 24th, with the exception of October 10th when it's fall break, then we won't have class. Please arrive on time. We'll get the space situation figured out, so we'll send out an update if we need to change rooms or have an additional room. Make sure you sign in. We'll pass in the roll sheet at the end of class. That's how we take track of attendance. And if you registered for the class so that it appears in your transcript, you do need to attend all the classes to receive a satisfactory grade. And if that is presenting any issue for you, see either Lisa or me after the class. We can talk about it. Occasionally, we do have assignments in advance of the class, reading assignments. So when we do, we'll send a notification. Please do read those and come prepared with questions. No homework assignments for today's class, so you can thank Dr. McLachlan for that. Nothing to do in advance for today. So. And our primary goal for the entire series is, of course, to answer your questions. So the worst thing that could happen would be for you to have questions and not ask them, to leave here with questions unanswered. So please ask your questions. We have a really stellar lineup of faculty presenters. So take advantage of their experience and expertise. Ask your questions. And if you think of things outside of class, email us or contact us. And we will answer you. So with that, I am going to turn it over to my colleague Lisa Wurtzel for our first exercise.
Thank you, Brian. So we want to try to introduce ourselves to each other. We're a pretty large group, so I'm going to ask that we do it fairly quickly. If you could state your name, what school you're in, your department, the degree you're seeking, and the type of research you're working on, that would be great. So try to keep your voices up so everyone can hear. And also, if you would be so inclined, maybe one fun fact about yourself, just as we're trying to get to know each other. So maybe we could start over here in this corner and then just come around the room. And if you forget the categories, just let me know. You don't have to stand up.
Medical sciences program. And I'm not in a lab yet, but I'm interested in the pharmacology department and endocrinology. And I look for old people.
Thank you.
Greetings, my name is Sunshine. My last name is Best. It is my real name. I attend School of Tropical Medicine and Public Health, and I'm in the Environmental Health Department doing an MSP in Industrial Hygiene. My research is related to retention and transmission of knowledge systems using mobile technology for the benefit of public health. And I love skydiving motorcycles.
Hi, my name is Blake. I'm environmental health, industrial health. Right now, I'm a clinical research assistant. I would like to do some environmental research for quality. I play drums.
I'm Kylie Brown. I'm in the School of Science and Engineering. I'm currently pursuing a PhD in cell molecular biology. Doing a lot of rotations right now, so I don't have any specific project in mind. I have no fun time. Just move on.
Hi, I'm Lucy Desmond. I'm postdoc in the physiology department. We work on neuroscience mostly. In fact, I'm French.
My name is Adrian. I'm a postdoc in the physiology department.
Okay. Hi everybody, I'm Melissa. I'm doing my PhD in the Department of Tropical Medicine, and currently my research is focusing on prediction modeling.
Hi, I'm Neil Wang. I'm a new PhD student in biomedical sciences this year, and I'm not that my rotation, so but I'm seeking my interest.
I don't know how to do that because I'm faculty.
Hello everyone, my name is Xinan. So I come from Tulane Medical School, and I'm a master student of Department of Safety.
My
name is McLachlan. I'm a professor.
Just make sure your son's on and you want to pass it
I
out.
received
At the end of the work form, I'll send the attendance certificate.
I'm Earl Murray. I'm
I was gonna see if I could get on it so I could get the emails for the, if you just email. Yeah. I can let you know. That's it.
Hi, everyone. I'm Bonnie Bowie. I'm a postdoc in the Center for Studies of the School of Public Health. And right now, my research is mostly on aging and health.
Well, I'm Trang Nguyen, and I'm a postdoc from the Center for Public Health.
Pass it out near the end of class.
I'm doing a research in aging and Alzheimer's disease.
My name is Brady Youngquist. I'm a BMS PhD student. I want to do like epigenetics research, and my favorite animal is a sloth.
My name is Tianfang Yang. You can just call me Frank, and I work in the cell molecular biology program. I'm a fourth-year PhD student in Tulane Uptown campus. I use mouse models to uncover the gene that is important for cardiovascular development. Fun fact is that I have a black belt in karate.
My name is Joe Berta. I'm a first year in the BMS PhD program. I don't have a lab yet. Interested in immunology and microbiology.
My name is Deep Chatterjee. I go by Deep. I just transferred from Florida State University, but technically I'm a first year grad student, PhD student at the BMS program. I work with Drosophila genetics.
If you come around here and then circle.
I'm Whitley. I got my master's in medical genetics here, and I'm doing research in cancer.
I'm Claire Pierce. I'm a first year BMS PhD. I don't have a lab yet, and I have to.
I'm Gemma. I started my PhD in neuroscience. I don't have a lab, and I'm doing my rotation system.
So hi, this is Ahmed Mustafa from the Department of Structural and Cellular Biology. Our research is on prostate cancer and inflammation.
I'm Javier Miller. I work at the same lab as Ahmed.
I'm Ning Duan. I was in the field with my and my research work is focused on gynecology tumors.
Okay, my name is Zheng Xiang. I work in structure and I work in the Department of Structure and Cellular Biology, and my current has focused on the cognitive degeneration.
My name is Mary, and I'm a first year BMS PhD student, and I don't have a lab yet. Fun fact, I like to play the piano.
Arissa Mercer, I'm also a PhD student, so I don't have lab either. I'm interested in microbiology and immunology, and I've lived in 8 states.
My name is Alex Merzick. I'm also a first-year PhD student in biomedical science program. Also not in the lab either, but I'm interested in pharmacology.
I'm Kunni. I'm a faculty in surgery department, and my research is focusing on thyroid cancer resistant development. I wanted to say my hobby is driving, but I recently had an accident, so I love to change that. I love to travel to different countries.
Hi, my name is Josue. I'm a first year BMS student here at the PhD student in the School of Medicine. Let's see. Fun fact. Research interests, developmental biology in pediatric area. Fun fact, from Texas.
I'm Jefferson Huggins. I'm with the School of Science and Engineering, Tulane's Brain Institute, studying neuroscience. And I don't have a lab yet either. I'm a first year, so doing rotations. And fun fact, I really like oysters.
My name's Deandra Ellis. I attend the School of Medicine for program of biomedical sciences. I'm a first year PhD student. I don't have a lab, and I like cooking.
Hi everyone, I'm Qiao Zhuxue. It's my first year PhD program in epidemiology. My research interest is nutritional and genetic epidemiology.
My name is Naomi. I'm a first year MPH student in epidemiology.
And yeah, I guess my fun fact is I'm binging Gilmore Girls right now on Netflix.
Yes. I'm Elise Facciati. I am 3 days into a PhD program in the Department of Aging. And I'll give you 2 really quick fun facts. Up until June, I was a financial advisor with Wells Fargo. And I have a 3-year-old grandson, and I bet I'm the only person in the room who can say that.
I'm Laura Lee. That's one of the wonderful tenured faculty. And one of my sabbatical projects is to evaluate how best to train in responsible conduct of research. So I'm here learning about that. And since I went on sabbatical, I picked up piano lessons again after more than 25 years, and that has really been a joy.
My name is Lauren Dion. I'm in the Aging Studies PhD program. My research interests are well-being, death, and dying, and existentialism. And my fun fact is I love Taco Bell.
I'm Mark Zimmerman. I'm stuck in the department. I'm also a master's student in clinical science, so I'm faculty and a student, so it's very weird dynamic. My research is sex hormones, therapy, and kidney.
All right, well, hello, my name is Sang Miller. I'm a first-year PhD student in the neuroscience department. So I don't have a lab yet, but I'm interested in learning about and studying about prenatal stress. One fun fact is that up until last month, I was in Kyoto, Japan.
Hello, everyone. My name is Justin McGrath. I'm a 3rd year in the MD PhD program, so first year of my PhD this year, working with Dr. Bunnell. My project is going to focus on using CRISPR to treat Krabbe disease. Fun fact is I like to whitewater kayak.
Hi, I'm Lexi Allen. I'm the first year veterinary resident at the Department of Comparative Medicine. So if any of you guys use animal models, I'll be seeing you guys. I'm recognized. Fun fact is I'm one of the few people that I've met so far that's actually from New Orleans. So fun being back home.
I'm Rita Richardson. I'm a research coordinator in the Department of Orthopedics from med school. And I actually have 5 labs, so if anybody wants to come play, I can find you something to keep you busy. And probably it's not necessarily fun, but it's weird is that dealing with cadavers doesn't bother me, so I've got a freezer full.
Okay. My name is Catherine Bunting. I am the director of the Standardized Patient Program at the School of Medicine. I am looking to do some curricular and academic research with my standardized patients. And fun fact is, in my other life, I'm an animal cognitist, and I work with large cats.
My name is Amy Mikoyevsky. I'm in the Department of Psychiatry and Behavioral Sciences. My research interests are primarily in the development of behavior problems in young kids. Fun fact, I have been a postdoc at Tulane until just July. I've got hired on as faculty.
Great.
My name is Katie Potts. I am a 2nd-year PhD student in the Department of Epidemiology. My research focus generally is on nutrition and lifestyle epidemiology and social epi. And my fun fact is that I do a lot of competitive sailing.
My name is Megan Allen, faculty in the School of Medicine in the Department of Pediatrics. And my research is on the impact of prenatal opiate exposure on newborns, especially nervous system. And I'm in the Physician Pipeline Scholar Program.
My name is Miranda Horn. I'm a first-year student in the neuroscience program rotation, so I don't know yet, but I'm interested in neurodegeneration, and I have 3 large dogs.
Okay, so if the folks in the overflow room here in JBJ can hear us, which I hope you can, if you all could just go around the room and introduce yourselves as well, please.
Sure. My name is Alex Rosado. I'm a 5th-year MD PhD
We're having a lot of trouble hearing you. If you could try to speak up really loudly. I'm not sure exactly where the mic is in there.
Okay, I guess I'll just restart. So my name is Alex Razavi. I'm a fifth year PhD student. My research focuses on cardiovascular disease risk prediction, particularly involving heart failure, but also sudden cardiac death. One fun fact is that I'm a big Buffalo Bills fan.
I'm Josh Bundy. I'm an assistant professor in epidemiology. I study cardiovascular disease and chronic kidney disease. Fun fact is I'm having my first child in October. I'm terrified, but it'll be good.
I'm Ellen. I work at the Tulane Cancer Center. I learned how to play the violin.
I'm William Robinson. I just started working at the Office of Clinical Research. And fun fact about me, I studied archaeology and undergrad.
My name is Austin Jones. I'm a fifth year MD PhD student in epidemiology. I study access to care for hepatitis C and chlamydia. A fun fact about me is I spent this past summer in Congo working the Ebola outbreak with WHO.
Okay, I'm Lydia Smith Osborne. I'm a first year PhD student, and I am still rotating, don't have a lab yet. Fun fact, I have a tortoise.
Is that everybody in the overflow room?
Oh no, there's Hi, I'm Stephanie White. I am a first year student, 3 days in. And fun fact about me is I was a Peace Corps volunteer recently in Madagascar. And my area of interest is behavior change as it comes to infectious diseases and preventing infectious diseases. Okay. Hi. My name is Shannon Escher, and I'm a postdoc in microbiology and in fungal infection. And a fun fact, my lab recently just moved to Tulane from LSU.
Hi, my name is McLachlan. I'm a first year biomedical science PhD student. I don't have a lab yet, but my interest is cardiovascular research. Thanks.
Hello, I'm Josh Curry. I'm a first year BMS student. Currently rotating, but my research interest is infectious disease. My fun fact is I don't like to play sports whatsoever. The opposite.
Is that now everyone in the overflow room?
Yeah.
Okay, so let's move to Uptown. Uptown, can you please introduce yourselves?
Can you hear us?
Yes, we can hear that.
Okay. I'm Kayla J. I'm the administrative assistant for Brian and Lisa, and y'all can contact me for any questions they have.
Okay.
Hi, I am Shoma Munshi. I am a postdoc fellow in the Tulane Brain Institute. Just that, I work with the investigating the electrophysiological properties of the inhibitory circuits in the amygdala and in its relation with alcohol addiction.
Hi, I'm Ann Mullen, and I run the transgenic core facility making genetically modified rats and mice for research.
I'm Savannah Stedman. I'm a third-year PhD student in biomolecular engineering, and my research focus is on polymer crystallization. I'm Susan Kelly.
David as well. Hi, my name is Shapeen. I'm also a person and my research is based on Hey, I'm Gloria. I'm also a first year PhD. Biomedical engineering.
Hi, I'm Anne Marie Womack. I'm a professor of practice in English, and I do research in disability studies.
Hi, my name is Vaughn. I'm also in the English department. I'm a visiting assistant professor, and my research is in the area of medical humanities as well.
I'm Connor Herring. I'm also a first year PhD. So I don't have.
That's it, Bobtail.
Thank you, Kay. Folks at the Primate Center, could you please introduce yourselves as well?
Okay, we're going to give this a try with our fancy new microphones in this room.
It actually sounds great, loud and clear so
well,
far.
I'm next to the good microphone, so I'm not using this I'm here at the Primate Center. I'm going to turn it over to our fabulous crew. See if you can hear you.
So Primate Center, we can't hear anything. Maybe you could just, each of you try to get closer to the mic to introduce yourselves, because we're not hearing anything right now.
Okay, we'll let our media team know that the new fancy mics aren't working. We'll have everybody come out.
Hi, I'm Julie Thompson. I'm a T32 veterinary fellow with the BMS program. I'm still rotating. I don't have an area of research yet, but I'm interested in public health and infectious disease. My fun fact is that The Walking Dead was filmed 5 minutes from my house. I am Sarah Kieser. I'm a veterinarian, the lab animal resident here at the Primate Center. I think my research is going to be something to do with monkeys and behavior and drugs. And fun fact, I was late because I was taking care of a baby monkey.
Hello, my name is Grace Wager. I am a PhD student with the BMS program. My fun fact is that I lived in 17 states growing up.
I'm Alex Nelson. I'm also a first year BMS PhD student, and my fun fact is I have a twin brother.
I'm Kathy Trumbull, a T32 veterinary fellow, still rotating. Fact is, I have 3 siblings.
And that's everyone here at the Primate Center.
All right, thanks. And so Primate Center, if you all have questions as we go through the presentation, please know that I guess you're going to have to come up to the mic to ask them because we're not hearing you when you're in your seats. So we want you all to participate, so we just want to be able to make sure that we can hear you. Okay. All right, well, thank you everyone so much. Thank you for introducing yourselves. I feel like we all know each other a little better now. And we had a big group, so I've got to get our speaker introduced so we stay on time today. We are very, very honored and privileged to have Dr. John McLachlan speak to us today. He received his undergraduate degree from Johns Hopkins, where he was co-captain of the varsity football team. So I guess that might be his fun fact. He might have another one to share with us. He received his PhD in pharmacology from George Washington University. Here at Tulane, he is the Weatherhead Distinguished Chair in Environmental Studies and also a professor in the Department of Pharmacology in the School of Medicine. From 1995 till 2012, he was the director of the Tulane Xavier Center for Bioenvironmental Research, which is a comprehensive center that deals with the environment in an inclusive manner. Before joining Tulane, he served as the scientific director of the NIH's Environmental Health Sciences Center. And while there, he developed the conceptual framework for what is now called endocrine disrupting chemicals. He has published more than 220 peer-reviewed papers and 60 review articles dealing with the environment and the reproductive system. His current research focuses on using the principles of hormone signaling to understand how factors as diverse as trauma, stress, heavy metals, and environmental chemicals exert their adverse effects on human health. And just this week, it was announced that Dr. McLachlan and his research associates were awarded a $1 million grant from the Keck Foundation to study how sex differences shape disparate immune responses in men and women. I don't know if any of you know much about the Keck Foundation, but they fund only extremely novel, cutting-edge research, the types of things that the NIH won't fund yet. So that is quite an honor, and we're looking forward to seeing what comes from that research. So without further ado, please help me welcome Dr. McLachlan, who will speak today about societal impact.
Thank you very much. Let me organize. Great. So, my name is John McLachlan, and a fun fact is the Keck grant she just bragged about was actually the work that was done by my son, James McLachlan, who's a professor in immunology, and he needed someone who understood reproductive biology and sex differences, which is something I've worked on for years. So he said, hey, Pops, can you give me a hand with thinking through this serendipitous finding he made in his own lab? And I said, sure, but if we're really going to go after this, we need one other guy who really understands sex differences at the molecular level. So we picked up a professor in the Department of Medicine. His name is Frank Movis-Javis, who's an MD, PhD, and he joined the team. And then between the three of us, we just kept putting together different approaches and sending it in. But it was all really based on a serendipitous finding he made in his lab. And I'm actually going to talk a little bit about the role of serendipity in doing research. So that's a fun fact. One other fun fact I was thinking about before she said that was that relates to your lives in the next couple weeks is I live in a loft on the 5th floor of an old office building at the corner of Magazine and Gravier Street in the CBD, close to the Sheraton Hotel, to give you some idea, on the 5th floor. I've never had a flood there, but I have never been in New Orleans when a hurricane came. So when Katrina came, I was in London. Any hurricane that's here, and I have to warn you, I'm leaving town tomorrow. I'll be gone all next week. So, good luck. So I'm going to talk a little bit about societal impacts as it relates to some of the topics you're going to be dealing with in terms of conflict of interest and how we share data, how we acknowledge authorship. So we'll go through some of these ideas. Society and research, as I'm thinking about this talk, and this is sort of an organization of the talk, we're going to be talking about research and its impact on society, and we'll do a lot of that and talk about some of the scope of that. But also I felt it was important, especially in this time of anti-scientific fervor, we explore the pushback of society on research. And then in the very last slide, I'm going to propose to you what I think are some of the attributes that might go along with a society of researchers. So we're going to talk about society in a couple different ways. Can you see it all? You want to lean over here against this podium? Huh? Okay. I'll describe it in such a way that you won't have to look all the time. Okay, so that's sort of where we're going to head. And we have to think about the whole idea of research as something that's been going on for centuries, if not millennia. And what we really need to know about research is you only know what you know. And what you know changes over time. And the context of research then is in that kind of knowledge base. And if we look at public health in the Middle Ages, let's say the 1400s or the 1300s rather, 14th century, this was sort of the public health approach to the bubonic plague. So here's Eric Idle, Monty Python and the Holy Grail. All they knew how to do was push a wagon through it, and this was actually something they said during the Black Death, the bubonic plague. They would push wheelbarrows through London and say, bring out your people, throw their dead, as they do in this particular movie. So it took a lot from that to get to where we are now and what we know in the context of your research, where we can talk about things like authorship. But again, it's important to know that knowledge changes, and the totality of knowledge is something that goes from time to time. We know that the Black Plague was a result of what? How did this all happen? What was the disease vector? Bacteria, what? But where was on
mice?
rats?
Rats.
So it was germ on a flea on a rat, and that actually came from Asia and ended up in Europe. So the whole idea of the whole idea of how these things geographically are distributed was taken advantage of by Dr. John Snow, who was a physician practicing medicine in London. And this is a famous story about an outbreak of cholera in the middle of London. And he was investigating how this could happen. This was in 1854. So this was sort of how we started to be able to take that diffuse, bring out your dead, and bring it to some sense where we could predict what to do next or anticipate what could happen based on research. And in fact, if you look right here, you see this thing says pump. What he did, these black X's are dots where people had cholera. Where were the cholera cases distributed? And he had to do this by himself and students going out and walking through the neighborhoods and interviewing people. So essentially, this is a map of disease before there was GIS and computers. It was all done by hand. And essentially, he's considered the progenitor of epidemiology because of the way he worked this out. The end of the story is, without really knowing why exactly it was happening, he didn't know what cholera was caused by. They took the handle off the pump on Broad Street, and this is Broad Street in London, not Broad Street here in New Orleans. And people couldn't get the water, and all these people who had been getting their water from a contaminated well, the disease went away. So that was sort of research without knowing what the cause is, but you still were able to make associations that had an impact on society. So here we'll move up a few years, and this is Louis Pasteur, which I'm going to come back to him again. But he's sort of considered, if not the father, one of the people who established the concept of microbiology and germ theory. And he started out as someone who was working actually in a winery, in a vineyard, and was able to show that by heating wine, he could keep mold and other microorganisms from not hurting the wine. And he called it immunization of wine. Then he did immunization of milk, which we now know is called pasteurization of milk. And then he was actually addressing an anthrax epidemic in a small village in France. And by trying different kinds of things, actually doing experiments, he was able to find a causal agent and even come up with an immunization strategy that stopped this spread of anthrax in this particular village. So in a way, we have to give him credit for pulling together the concept of germs and the germ theory. And once we had a germ theory, we could make assumptions and make moves to how to look at disease in its broadest sense. As an environmental scientist, I'm still frustrated that we don't have a germ theory of the environment. We don't have any causal link. It's too diffuse. And so we can't really say, oh, if we could do this, this would happen. So we continue to make these kinds of semi-causal associations. Whoops. So in the attempts to pull these things together, in the 20th century, research became an increasingly integral part of society. And it really, as I read this in preparation for this, pulling this talk together, I think all of these slides except 3 are brand new. They're all sort of thoughts for this. Is the conduct, World War II was really a factor that focused research in the United States especially, and it pointed out the need for research coordination. And 2 examples is atomic nuclear research, the atom bomb, all of the nuclear research that was going on in the national laboratories and universities were focused and then brought into what was called the Manhattan Project, whose endpoint was to develop a weapon that was so lethal it would stop World War II. Ultimately, that was the fission nuclear bomb that was dropped on Hiroshima, but that required a tremendous amount of resources and coordination. The other example that came to me as I was reading this was the advent of antibiotics. And Alexander Fleming discovered penicillin totally serendipitously. He was, I don't know how careful you all are in the lab, but he had a reputation, from what I've read, of just being a little bit sloppy in the lab as a technician. So he was growing some bacteria and went off for a vacation and left some of the dishes on the covers on the petri dish were ajar. The mold got in. When he came back, his technician said, "There's all clear stuff here." And he looked and, oh, there was something growing. It turns out that mold was making penicillin. This was in 1928. By 1942, the first private patient was treated with penicillin for a woman who had septicemia after miscarriage. And by 1943, it had a clinical trial and didn't have a clinical trial. It was actually being used in the war to prevent death and amputations for people who were wounded. And when the war started in 1939, there was no penicillin. 42, they discovered that it worked. 43, there were about 4 million units of penicillin. It was still rare and hard to use. By the end of the war in 1945, American industry was making 6.5 billion units of penicillin every month. So you can see how these things can go from basic to impact in a short time. And so the issues of applied and basic research after the war were debated and are still debated, and that's sort of where some of the stuff we're dealing with now comes to. And a book that I would urge you all to read, that I read some years ago and I keep rereading, is called Pasteur's Quadrant. It's been pointed out to me by my partner in life that I don't pronounce Pasteur correctly, but you know by reading it what I mean. This was written by Donald Stokes. It's a very thin little book, and Donald Stokes was a professor in the Woodrow Wilson Center at Princeton University, and his thought was, how do you integrate basic science into the overall scheme of things in a way that makes some sense? Yes. So one of the things he points out in the book is that Vannevar Bush, in a report he wrote in 1945 at the conclusion of World War II called Science, the Endless Frontier, concluded that applied research invariably drives out pure. In other words, basic research is at risk because applied research, you can find something out right away and apply it, and basic research might take decades or multiple decades to get from what you find in a dish to what is actually saving lives during war. And so he was, during the Second World War, he was the director of the Office of Scientific Research and Development, which actually pulled together the Manhattan Project, made computers, developed encryption, took care that penicillin got in as antibiotics. So this office was coming to a conclusion at the end of the war. And so his instinct that something needed to happen for basic research actually led to the formation in 1950 of the National Science Foundation. And then the National Science Foundation has funded fundamental research ever since then. And the National Science Foundation pushed the National Institutes of Health to focus on basic research as part of what they did. So you can see the impact this kind of thing has. So in the line of thinking that was outlined by Vannevar Bush, is you have basic research gives rise to it, is then picked up and applied by others, developed by yet others, and then there's production and operations. And what Donald Stokes, Professor Stokes, talks about is a new model, a different model that I really like a lot, called the quadrant model of scientific research. And again, this is from his book in 1997, in which instead of having a linear array of research, you look to see what inspires the research. Is it use or is it fundamental understanding? And so we can look here at pure basic research, which he calls Bohr's quadrant, named after Niels Bohr, who was trying to understand how particles went around in atoms, and he didn't care that everything he was looking at was ultimately going to be nuclear power plant or an atom bomb or something else. He was only interested in how did that work at that fundamental level. Down here, where there's no interest in fundamental level theoretically, and all consideration of use is called Edison's quadrant, because even though Edison had probably more patents than any other American ever has had and generated tons of new information about electricity, his main driver for the research he did was to electrify the United States, make light bulbs, and sort of create an industry. And so this is what would be called Edison's quadrant. And then up here in what people know is the sweet spot. Anytime you do a quadrant analysis, you always know that whatever's in the upper right is what you should say, oh, that's what I want to be, or oh, yeah, that's where I am. So this is called use-inspired basic research, and that's named after Louis Pasteur, who actually created, in some ways, and I hate to say all this, having experts in infectious diseases in the audience, Dr. Levy, most among them. What's interesting, Laura, is I hardly have any endocrinology in this. Once I got going on impact, I became a microbiologist and an infectious diseases and a vaccine person, as you'll see in a minute. Yeah, right. So it's taken a lot of checking with people. So this is sort of this area of use-inspired basic research. And as an example of that, I wanted to bring in somebody that I actually knew. And this is Martin Rodbell, who worked at the same institute that I did, National Institute of Environmental Health Sciences. In fact, this is a picture of Marty at a celebration for his Nobel Prize. And this guy is Nick Carter, who was the first scientific director at the institute when I got there. And then when he stepped down, Marty took over as the scientific director. And then when he stepped down, I took over as the scientific director. And what Marty was thinking about was how do cells transduce signals? And this figure is taken from his Nobel lecture. I would encourage you, this was 1994, I would encourage you as young scientists to just go online and Google a Nobel Prize winner or a topic, and they all have a Nobel lecture that's in a book or usually published in Science magazine. And these guys, I swear to God, they know they're going to be famous. So they write stuff when you're your age doing your stuff, and then they refer to it in these Nobel lectures. Oh, yes, when I was 28, I thought I would do such and such and such. So you read this stuff about Dr. Rodbell, and it really gives you an insight to how he thought. And so here's basic science. He's actually looking at a cell membrane as a kind of electrical switch. He was a radio man in the Pacific. He was an 18-year-old Marine during the Second World War. And he started thinking, how could I take a radio analogy and put it into a cell membrane? And as you know, he's got the Nobel Prize for discovering G proteins. And in terms of application, this is just a cover of a issue of Science of a couple years ago, where you look at this signaling, not that his work on G proteins led to all of this, but it was one of the signal events in signaling. And now there must be 1,000 drugs that are related to cell signaling. Hypertension, schizophrenia, pick a disease, there's a cell signal that has something to do with that. So clearly it was impactful, and I thought the juxtaposition of these were so dramatic, and his work, I think, really points that out. In fact, here's one you don't have to look up. Just look up Martin Rodbell, signal transduction, Nobel Prize, 1994. So if we come back again to this Pasteur's quadrant, when I was talking to a friend of mine, Bill Toscano, who's a professor in Minnesota, about this talk, this is a while ago, and we were talking about this, and he had read this book too, and he said, you know, if this basic science, use-inspired research wasn't operational and everything was pure applied research, the response to the polio epidemic, polio was infantile paralysis, poliomyelitis, it was rampant in the first half of the 1900s of the 20th century. And one of the things you could do is, one of the outcomes is you would have paralysis of your muscles. And if it was your legs, that was called spinal version of polio. If it was your throat and your chest, that was called bulbar, and when your chest muscles were paralyzed, you couldn't breathe. And they would put you in a machine called an iron lung. And so he said, if it was just applied research, if we didn't have this side of it, we just have better iron lungs now. And that really struck me. And I thought about that. And so here I've sort of explicated that. Here's an iron lung up here, and I'll come to that in just a minute. But in 1952 and 1953, there were 58,000 and 35,000 cases of polio in the United States, of which in 52, 3,000, mostly kids, died, and in 53, 1,400, mostly kids, died. And this is personal with me because I got polio in 1953. I was 10 years old, and I spent months in the hospital, almost got in an iron lung, ended up with just a brace and a crutch that I had for about a year. But when you come back and look at this, patients with breathing difficulties were given assisted respirators. And here's a child in an iron lung. They're essentially whole body tubes that have negative air pressure and positive air pressure that breathe for you. And there are cases of people who have spent 30 years in an iron lung. And in one case, in Bill's vision, it would have been, and these actually existed, iron lung farms working better and better. But because of the way science can go, there was a use-inspired basic research. Jonas Salk described a vaccine in 1952, and in 1955, a giant clinical trial concluded, and this says here, Salk's vaccine works. And so this is 55 newspaper. Here's 1957. This is in a special building on the University of Pittsburgh campus called the Cathedral of Learning. And fortunately, Jonas Salk worked at the Children's Hospital in Pittsburgh, and he's in here. I tried to see if I could see him, but according to the legend where I got this, Jonas Salk is one of the physicians from the Children's Hospital of Pittsburgh administering vaccine to hundreds, thousands of kids who started getting that in 50, this is in 57, and by 94, polio is eliminated in the Americas. So this is a success story, a big impact, but we're always our impact is always being modulated by the community, the society we're impacting is impacting back on science. And that's something you need to know about and you need to think about, and you need to be very careful about how you do your science so that pushback doesn't find chinks and gaps in what it is you say. So, again, I showed this talk to my son, James, who is an immunologist. And he was, Dad, I can't believe it. You become an immunologist and microbiologist for this talk. So this actually, he gave me this slide. So this, in the pre-vaccine era, talk about societal impact of research. Here's the estimated annual morbidity in the U.S. These numbers don't show up as well as I'd hope, but they're in the tens of thousands, hundreds of thousands, this case 4 million. And if you just look at these numbers, these are the percent decrease after vaccines, and they range from 100% to 90% to 80% to 85 to 90. So vaccines have had this enormous impact on society and on health. But where measles, I think, is right there. Is that right? Mm-hmm. Measles. So this number is bigger now, and I just wanted to point that out. So in terms of impact, maybe antibiotics have had as big an impact. Fluorination of water maybe has had that kind of impact. But I think vaccines have to be given credit as a hugely impactful research. But now I want to use this as an example, as how I mentioned before, where society pushes back on research and how we have to use all the things you're going to learn during this course to make sure that the pushback is muted as much as possible. So we can talk about irresponsible conduct of research and the generation of societal uncertainty. And I'm going to use the word uncertainty in the next 2 or 3 or 4 slides. And here it's the mumps, measles, or the measles, mumps, rubella vaccine as a case. There has been a kind of anti-vaccination movement in the UK and America for years before this happened. And some of it's for, in terms of many precedents, some of it's religious reasons, some are other kind of reasons. But a sharp stimulus in the 21st century was the claimed association between the MMR vaccine and autism in children. And this was a paper by Andrew Wakefield that was published in The Lancet in 1998, which is now a notorious publication. It's been analyzed thoroughly by peers, by panels. The British Medical Journal in particular did a 3-year analysis and determined that this is a fraudulent research study. There's no doubt that it's scientific fraud. It also has been identified through email strains that Dr. Wakefield had a financial conflict of interest and himself was conspiring with companies to make his own diagnostic kit that would pick up the things that he was claiming were happening. This was retracted in The Lancet, but it took 12 years, 2010, and it was retracted by the editors of the journal, not by the author himself. So this is another example of, if there's a mistake and you make it, correct it. Yes?
Can you take a photo?
Sure, yeah, all these slides will be on their website.
Oh, okay.
Okay? Everything I'm saying will be there. Not everything I'm saying, but everything that I'm putting up will be there.
Why did it take so
long? It was tricky at first, and I think people tried to get him to do something about it, and he wouldn't. He eventually ended up losing his medical license, lost his job, lives in infamy. But look at this, but that's 12 years of this has been cited. Okay, so uncertainty in research then becomes an issue that we have to grapple with. As scientists, we have uncertainty. That's sort of how we do our work. We use statistics to say, oh, that's certain standard deviations, or my confidence interval says I'm 0.01 or 0.05. You all know those kinds of things that we say, highly statistically significant, whatever that means. But it's something that gives us a sense of certainty and uncertainty. In all of science and research, there are only some findings that I think are absolutely certain. And the best example is the Earth goes around the sun. But that took centuries to establish, and a lot of controversy, and people had worse than having their papers retracted. They were burned at the stake and, put in jail, all sorts of terrible things. That doesn't happen now, although you can imagine that something close might be happening. We'll get to that. So one of the places where I want to bring this to your attention as researchers is that over the years, special interests have used the concept of uncertainty to create societal skepticism towards science. And here are 3 well-known examples. One is tetraethyl lead in gasoline. So in the 1930s, lead was added to gasoline as an anti-knock agent. Everybody knew that lead was terrible. Everybody knew that lead was a neurotoxin. Everybody, going back into the 1800s, it was said lead is a poison. Lead-based paints, if you're painting, don't get it on you, you'll get sick. So everybody knew that, and yet when they brought up this as an issue, there was an uncertainty measure that was drummed up by special interests, in this case gasoline and the tetraethyl lead companies, that said, no, we really don't know. It really might not happen. They finally, a president who I didn't have a lot of respect for, Richard Nixon, signed the Clean Air Act in 1970 and followed this up with a Get the Lead Out of Gasoline bill. And by the 80s, lead was out of gasoline by the mid to late 70s. And to give you an idea of how important and what an impact it had, from the time in 1970 to 1990, the level of lead in kids in the United States has dropped 80%, and the IQ, average IQ, has gone up 3 to 4 IQ units. And that can actually be calculated by economists in terms of gross national product and what that can mean to a country that's essentially poisoning their kids and lowering the overall IQ. And it isn't just the middle of the bell curve. It means there are more kids who are real slow and fewer kids who are gifted. I was talking to somebody today as I was going over this slide, and I thought, man, I would have really been smart if I'd been born later. The other is cigarette smoke and health. Nobody smokes. Nobody likes to go in a restaurant where you smoke. Somebody might smoke, but you know if you smoke, it's bad for you. You're going to get lung cancer. You're going to have emphysema. And yet, some of the same organizations, even the same public relation companies, there's a whole book on this, got together and created uncertainty in thinking about cigarette smoke and health. The same public relation companies have been working for decades to say, we don't really know the global climate change. So you talk to these scientists, they'll never say it's conclusive. They'll say it looks like it. So that creates uncertainty. And so the same approach is being used here. Societal impacts of global climate change research are profound. And these are just, we could have a list that goes the whole way down and goes over up onto your shirt. But here are just four that I think of right away. And I teach a class called Environmental Signaling in Medicine, where we end up having to go to the board and everybody has to figure out, okay, global warming, what's it going to do? But emerging diseases, certainly disease vectors, clearly happening. Sea level rise, coastal city loss, that means mass migrations. Millions of people are going to be displaced and go somewhere. These are all just health related. Disruption of agriculture, fisheries, food supply, decreases in water resources. This one, emerging diseases, actually a grant that I stimulated Don Wesson, who's a professor in the School of Public Health, in 1995 to write a grant called Emerging Diseases in Changing Times. When I was a center director, I knew to get grants, the first thing you needed was a good title that would have a good edict, we called it. Oh, everybody wants a fun edict. She actually predicted that as we became more and more tropical here, emerging diseases would show up like dengue fever. And at first there was, oh, that would never happen. So now there are cases of dengue fever in New Orleans and Houston. So I just wanted to give a shout out to her and that grant that NIH being one of the first in this area. So we can, if we do careful research, we can lessen uncertainty. And coming back and sticking with global climate change, a recent paper just in February 25th of this year, analyzing the three major data sets which get analyzed and reanalyzed, concluded in Nature Climate Change that there is an association between human activity and an increase in temperature, and it reached what's called the golden level, the 5 sigma level, which is, in terms of p-value, is a probability of 3 times 10 to the minus 7, or you end up with 1 chance of a mistake in about 3 million tries. So this is approaching certainty, but that's the whole problem. As a scientist, you can never say the Earth goes around the sun. You know, you'd say it's really, really good. 5 sigma, hardly anything gets at that level, but this is sort of tells us where we're headed. I'm almost done. So, ultimate uncertainty. This is a cartoon by our editorial cartoonist in the Times-Picayune, Walt Handelsman, who's great. And it's when I saw this title, the Science Deniers Club, I thought, oh man, but you're going to be dealing with this. You're going to be coming up, explaining your work, trying to communicate with the public, and you're going to have people who are, denying science, although in this case they'll get wet. And so finally, I think this is my last slide. Pardon?
Okay. There are multiple good points. We just weren't sure.
Oh, okay. If you need a break, we can do that. But so in terms of society of researchers, responsibility and values, anticipating what you're going to be hearing in terms of conflict of interest and authorship and conduct and everything, I thought through what do we expect for people joining the community of science and research. And so certainly, at the top, you'd expect integrity, transparency. You want people to know what you're doing and why. Fairness, that solves authorship problems. If you're fair and you include the people who did the work in the right way, then you don't have to have that many rules, although, as we know, you do. Honesty, inclusiveness, and this one I put in anticipating the whole ideas of looking at sex differences, including female subjects and animals, looking at difference in race, in ethnicity, in human studies. I mean, I've reviewed papers where people try to report on the effects of hormones on menopause in male rats. It's easier to handle male rats. They don't have an estrous cycle. And then in the last one, be careful in what you do, but don't be catatonic. I've talked to people who are like, oh, you know, if I publish this, it's so unexpected, it'll probably be wrong. And I said, well, but don't, you know, be careful, replicate it, reproduce it, be sure of what you have, but then go ahead and do it. You know, that Keck grant that was mentioned, my son James, who had the work, it actually occurred as a serendipitous finding, a little bit like Fleming's, in his lab, and he didn't know what to make of it. And it sort of got around, okay, I'm willing to put it out and see what other people think. So don't let some finding that seems unusual slow you down, but check it. And I'm not going to do this. So that's the last slide. And is there time for questions? Yeah, if you have any questions. Okay. Well, I think everybody wants to go because the Saints are going to be at the Superdome, and it's going to be hard to get out of here in 5 minutes.
And so we had, again, apologies about the room. Look for it's on the roll sheet with your email address. Look for an update about the room. And we had some logistical issues in the beginning. Normally we'll have more built-in time for a discussion and questions, but we're thrilled about the level of interest. Again, this is most people we've ever had attend. So thank you.
Congratulations. Okay, thank you. Right? Okay. You're welcome. Was it close to what you wanted?